All righty, why don't we go ahead and get started since it's noon. I am Teresa Smolarek from Cincinnati Children's, and I wanted to welcome you on behalf of the Education Committee of the Cancer Genomics Consortium to our next webinar. As way of a housekeeping detail, please put your questions in the Q&A, and we will get to as many as we can during our time. And a recording of the webinar will be posted to the CGC website afterwards. And I would like to now turn it over to Gordon Aratza from Children's Hospital of LA to introduce our speaker.
Thank you very much, Teresa, and thank you everyone for joining us today. It is my great pleasure to introduce our today's speaker, Dr. Zhaohui Gu, who is joining us from City of Hope. Dr. Gu is an assistant professor in the Department of Computational and Quantitative Medicine and Systems Biology at Beckman Research Institute of City of Hope. He earned his PhD degree in bioinformatics and cancer biology at Shanghai Jiaotong University and did his postdoctoral training at St. Jude's Research Hospital, where he was the key contributor to multiple large cohort studies that use transcriptome sequencing, genome sequencing, and advanced analytic approaches to refine classification of pediatric lymphoblastic leukemia, and also to define several new molecular subtypes, bringing the number of different subtypes of leukemia to more than 20. And at his current position, Dr. Gu continues to focus on a molecular basis of high-risk BLL, as well on perfecting further his analytic approaches to refine classification and understand molecular pathogenesis of leukemia. In addition, his laboratory focuses on one molecular subtype that was discovered through his previous research, which is PAX5 alt BLL, understanding molecular pathogenesis of PAX5 alt. Dr. Gu's research has been recognized and supported by the ASH Scholar Award, Leukemia Lymphoma Society's Special Fellow Award, and NIH NCI Pathway to Independence Award. And then finally, I have to say that here at CHLA, we had a huge privilege to collaborate with Dr. Gu to work on applying some of his advanced analytic methods to deconvolute some of our interesting and difficult BLL cases. And I have to say that has been very interesting, very educational, and really fun. So I really hope we can continue this collaboration and expand it in the future. So thank you, Dr. Gu, for joining us today, and we're very excited about your talk.
Thank you, Gordana, for the kind introduction. Also, thank you, Teresa, and the organizers for this meeting. It's really a great pleasure for us to present our research here in this webinar series. As you can see from the title, I'm going to talk about the different BL subtypes and the signatures we've been studying over the past few years. Let me start. Okay, I guess I'm just too junior to have anything to disclose here. For today's talk, I'm going to start with the background of leukemia, and I'm sure you probably know this very well. Then I will touch a little bit about the, the definition of leukemia subtype, because this is the basis of our research. Then we'll dive into the discovery of the novel subtypes, which is the main work I've done during my postdoc training and also the main work of our current lab. Then I will talk about some of the signatures we have been observed from some of the novel leukemia subtypes. I will start with this photo of Dr. Jack Miller, because he's the one proposed the idea that lymphocytes should be categorized as T cells and B cells. But at his time, he's been widely criticized for making lymphocyte or immunology too complicated. But now we're proposing the idea that leukemia or B cell acute lymphoblastic leukemia might have over 20 different subtypes. And hopefully will not be criticized too much because of this effort. Okay, leukemia, as we know, it's the most common pediatric cancer. For kids younger than 15 years old, leukemia takes around 1/3 of pediatric cancer. Even for young adolescents, leukemia is still one of the major cancer types. In the US alone, over 3,000 kids are diagnosed with leukemia. Over the past 50 or 60 years, the treatment for pediatric leukemia has been improved significantly. As you can see, here's the total therapy trials in St. Jude. The clinical outcome has been improved significantly. Now, over 90% of the kids can reach long-term survival, but still 10 to 20% of the kids, they would relapse, and the outcome for them is still dismal. That is why leukemia now is still a leading cause of childhood cancer death. Pediatric leukemia, there are mainly two subtypes defined by the lineage of the leukemia. One is acute myeloid leukemia, which is a minor subtype of pediatric leukemia. The other one is acute lymphoblastic leukemia, which takes around 80%. Within ALL, it can be further divided into T-cell ALL and B-cell ALL or B-ALL, which is a major group of pediatric leukemia. And our focus is in this subtype of leukemia. With a lineage-based leukemia classification, they can be further divided into smaller groups or subtypes, like FAB classification system, which has been developed a long time ago. And also it has helped the development of target therapy. So for acute lymphoblastic leukemia, there are 3 major subtypes. And for acute myeloid leukemia, there are 8 different subtypes. And here this M3, which have been later confirmed with the fusion PMRARA, and the legendary treatment, Ultra plus ATO, has been proven to be very effective treatment, and which made this leukemia subtype the first curable leukemia. It signifies the significance of doing the disease classification, because with this knowledge, we would know a specific treatment would be effective for a certain subtype, which will lead to the development of targeted therapy. In addition to the FBB system, which is mainly based on morphology or the immunophenotype, there are some other assays or platform have been used to further categorize the heterogeneity of leukemia. For example, in ALL, researchers and clinicians have been using cytogenetics-based or FISH assay to identify the driver genetic lesions in ALL. Even before high-throughput sequencing, there are multiple ALL subtypes have been identified, such as the hyperdiploid or hypodiploid, which are defined by the total chromosome number. And the ETV6 chromosome fusion, KMT2A rearrangement, or previously known as ML rearrangement, TCF3 PBX1 PH chromosome. This results BCR-ABL1 fusion. With this molecular subtyping system, different patients might be categorized with low or high risk, such as hyperdiploid and ETV6 chromosome 1. We know they tend to have very good outcome, while KMT2A, PH, and hyperdiploid cases, they tend to have very poor outcome. After entering the high throughput sequencing era, multiple novel subtypes have been identified. This is a pie chart showing the different AL subtypes. Here, the blue color indicates the one in BLL, and the orange color is, or the yellow color means the one in the novel subtypes in BLL. Here, the red color are the TLL subtypes. And here's a group of cases called other B, which means they don't have a specific subtype. Among all these subtypes, I want to bring your attention to this one called a PH-like or BCR-ABL1-like, which means they don't have the BCR-ABL1 fusion, but they have very similar gene expression profile. Among this subtype, multiple kinase activating fusions have been identified, which led to the discovery or the development of kinase inhibitor therapies to these patients, and multiple clinical trials are still ongoing. But before moving on, we need to have a clear definition of a leukemia subtype. So let's take a look at this gene expression profile, which is done in over 300 pediatric ALL. You can see here, different leukemia subtypes are grouped in this gene expression profile. And each one is mainly defined by the driver fusions or aneuploidy. So to give us the idea, each leukemia subtype, they share the similar genetic alteration, and also they share a similar gene expression profile. So using this strategy, in this study, which was done in 2002, they observed a very tight cluster here, but they cannot identify what was the driver genetic lesion. So they coined this one as a novel subtype. Later, with the SNP array-based copy number variation analysis, they observed many of the cases here, they have ERG intragenic deletion. So they call this subtype as ERG subtype, which some of you are probably familiar with. In 2016, a Japanese group, they identified all these cases here in this group. They actually carry the DUX4 gene arrangements, commonly with the IGH gene region. So officially, this subtype is called a DUX4 rearranged subtype. So as you can see, the definition of leukemia subtype, sometimes it's not that simple. And I will talk about this DUX4 subtype later in the following slides. Hopefully with that information, you get a general idea about the definition of a leukemia subtype. So first, on the genomic level, they should share same or similar genetic alterations, or they share similar gene expression profiles, such as the PH-like subtype, and they are enriched with a certain pathway mutations. Then on the transcriptional level, they should share similar gene expression profile, and if that, and because of that, the leukemic cell, they may respond similarly to a specific drug treatment, and consequently, the patient may have similar clinical outcome. So the disease classification is not just for pure research, and it's also closely connected with their clinical treatment and their outcome. Following that, the disease classification pipeline, I'm gonna walk you through quickly the discovery of the MAF2D subtype, which was my first project when I started my postdoc training. I started with analyzing a large cohort of BL cases, which consists of over 500 BL samples. After calling the fusions, I identified 22 cases that carry the MAF2D fusions with different fusion partners. Then I checked the gene expression profile. As you can see in this 3D PCA plot, all the MAF2D cases, they form a very nice gene expression cluster here, indicating they activated or repressed certain genes. Then we want to confirm what is the incidence rate of this disease subtype. So we carried out a targeted screening in over 1,000 BL cases and eventually confirmed around 3% of the pediatric leukemia should belong to this MAF2D subtype. MAF2D subtype, why we call that a MAF2D subtype? Because its fusion partners are different. Most commonly, MAF2D is rearranged with the BCL9 gene. Because of there are different fusion breakpoints. So there are, we call that a MAF2D BCL9 with 4 different S forms here. And all of them have been confirmed using RT-PCR and a FISH. And here also some other minor fusion partners with MAF2D also observed in the study. For the cases with the SNP array or whole genome sequencing data, we also analyzed the copy number variations. And as shown here, you can see around the MAF2D, there are multiple interstitial copy number gain or losses. Here, if the signal goes up, which means it's copy number gain. If it goes down, which means there's a loss. Because of that, you can see there's a gene, BCL9. It's very close to the MAF2D, which can partially explain why MAF2D is most commonly rearranged with BCL9. And also, because this event is within one chromosome, and it's very hard to be identified using the conventional cytogenetics-based screening. This subtype, after the discovery of that, we also checked the clinical outcome. And here is a COG AALL 0232. This is a high-risk group of patients. As you can see here, the MAF2D patients are showing with even worse clinical outcome compared to the KMT2A subtype, which is already a very high-risk leukemia subtype, indicating the MAF2D is also a high-risk leukemia subtype. So the treatment for this group of patients also needed to be improved. Then we checked the gene expression signature of this disease subtype and observed HDAC9 is significantly upregulated. There are multiple HDAC inhibitors available. So we did a preliminary drug screening or drug toxicity assay in a few patients. As you can see here, The panobinostat, which is also a HDAC inhibitor, are showing with very high sensitivity of killing the leukemic cells, indicating the HDAC inhibitor could potentially effective for this disease subtype. But again, this is a very preliminary study. More follow-up research are still needed to confirm if this could be a beneficial addition to the therapy. Well, as you can see, using RNA-seq, we can identify the fusions, and also we can evaluate the gene expression, and indicating the RNA-seq is a very powerful tool to identify the novel BL subtypes. So over the past few years, multiple leukemia or BLL subtypes have been identified using RNA-seq. Even with the discovery of these novel subtypes, still around 10 to 30% of the BL cases cannot be classified. So I came to my second postdoc project, the effort to identify all the novel subtypes in the rest of B other cases. To achieve that, we want to collect as many samples as possible because we're expecting some of the leukemia subtypes could be minor. We need to have a large number of patients to identify them. Eventually, we accumulated around 2,000 BL cases across different age groups and different risk groups as well. And here we proposed to do the integrative genomic screening, which means all the cases here, they will have RNA-seq to detect the fusions and their gene expression profile. And for the novel clusters, novel gene expression clusters, we'll perform whole genome or whole exome sequencing to identify their somatic mutations, including SNV and indels. And also using a whole genome, we can also identify the structure variations and the copy number alterations. Around half of the cases in this cohort, we have the SNP array data, which can be used to identify the copy number alterations. But before we move on to identify the novel subtypes, there are two issues we need to resolve first. There's one subtype I mentioned before, the DUX4 subtype, which is driven by the IgH DUX4 fusion. But this fusion is very challenging, very tricky to be detected because both IgH and the DUX4 are in the highly repeated region. Most fusion callers cannot identify this one sensitively. Another issue is the missing karyotype. In our cohort, around a quarter of them, they don't have the karyotypic data, which means we cannot classify the 3 aneuploid subtypes defined here. So for the first issue, the IgH DUX4, as you can see in this figure, DUX4, it is very close to the telomere region. And in human genome, there are dozens of DUX4 copies. They're highly similar. And again, the IgH region, it's also a highly repeated region. After inserting the DUX4 gene into the IgH region, which is highly active in B cell, which would result in the overexpression of DUX4. DUX4 itself is a transcription activator. And because of the activation of DUX4 and its following genes will be highly activated, which would give the DUX4 subtype a very unique gene expression profile. You can see here is a very tight cluster indicating they belong to the DUX4 subtype. All this discovery of the DUX4 subtype was, well, the major discovery was made in 2016, 3 papers published to describe this novel subtype. The conclusion here is this subtype has very unique, very specific gene expression profile, but detecting the fusion is very tricky. So what we did in our study is to do the analysis in the other way around. We do the gene expression cluster first to identify the cases potentially belong to this subtype. Then for the ones we do not see IgH DUX4 fusion, we would go back to check the raw alignment, the sequence alignment as shown in this IGV plot. You can see here, this is DUX4 gene. Here is the alignment of the reads. Partially, the reads cannot be aligned to the region, shown in the soft clipped way. If you zoom in, you can see the DNA sequence here. So what we did is to put all the sequence or assemble this sequence together into a longer contig, then align the sequence back to the reference genome. After doing that, we can see there should be an IgH DUX4 fusion. But because of this known sequence inserted in the middle, which made the detection of this fusion even more challenging. Because of this issue, currently in our lab, we're also trying to develop the GEP or gene expression profile guided fusion or structure variation caller. The concept is that first we do the gene expression analysis. We identify the cluster of each patient. If it belongs to a specific disease subtype, we would know there's some candidate genes we're expecting. Then we'll do the assembly-based fusion calling or the structure variation calling. The structure variation here would include the commonly affected genes such as FLT3, ITD, or KMT2A PDD, and also some intergenic amplification events here. Because we know some of the fusions are expected, we would score the fusions or genetic lesions identified in this process. And also we're gonna use our large BLL RNA-seq cohort to rule out the false positive result. This is an ongoing project which is led by a PhD student just joined our lab recently. Okay, another issue is the missing karyotype. As I mentioned, all the samples included in this cohort, they have RNA-seq data. So we're wondering if we can use RNA-seq to call the karyotype. So the idea is very straightforward. If there's a whole chromosome gain, we would expect the genes on that chromosome, their expression level should be elevated. And if there's a whole chromosome loss, the genes on that chromosome, the expression level should be lower. So we made this plot here showing the gene expression level across the genome. Each dot represents a gene. You can see some of the chromosomes showing elevated expression than the others, indicating it's probably a copy number gain. But to confirm that, we also need to find the baseline. The baseline means the chromosome has 2 copies. So we also included the mutant allele frequency of the heterozygous SNP. As you know, for the diploid region, if that is heterozygous SNP, the mutant allele frequency should be around 0.5. And if you make the density plot, you would see there's one peak around 0.5. But if there's copy number alterations, the mutant allele frequency will be imbalanced, and that will be presented in this 2-peak pattern. So combining these 2 sites of information, we can reliably call the whole chromosome number, copy number alterations. This is just an example shown here. This software is developed through collaboration with an MD student when I was in Dr. Charles Mulligan's lab, and it's just published earlier this year. And it's freely available from GitHub, and you're welcome to use. So in addition to call the whole chromosome level copy number alterations, this software can also sensitively detect the IAMP21. MP21 stands for intrachromosome amplification of chromosome 21. As you can see here, on chromosome 21, the gene expression level is significantly higher than just the 3 copies. And the density plot of the mutant allele frequency is also different than 3 copy. This indicating that's probably an MP21 event. Okay, after resolving the missing karyotype cases, we can move on to identify the novel subtypes within this cohort. Here, I would like to start with this t-SNP plot because this is so commonly used for single-cell RNA-seq data. It's very powerful to cluster the cells with similar gene expression profile into distinct clusters, as you can see here. Here, since we have around 2,000 BL cases RNA-seq data, so we can infer their gene expression and map them into this t-SNP plot. As you can see here, the well-established BL subtypes form very nice clusters. And in addition to them, we observed some subtypes or some clusters we have never seen before, such as this one, PAX5P80R. At the very beginning, we don't see any consensus fusion genes because fusions was originally we're thinking they're probably drivers. Then we went back to check the RNA-seq data to see if there's any sequence mutations. And surprisingly, all the cases here, they carry the PAX5P80R missense mutation, indicating this is probably the driver of this subtype. In addition to that, we found a close by cluster here. The majority of them, they carry different types of PAX5 alterations. So we named them as a PAX5 alt subtype. And here, another group cluster here. Most of them, they carry BCL2 or MYC fusion with IDH gene. So we call this as a BCL2 MYC subtype, which is very similar to the double hit B-cell lymphoma. Another subtype is a very minor subtype. All the 8 cases here, they carry IKZF1 NY59Y missense mutation. So since that is very tight cluster and all of them carry the same mutation, we believe this should be the driver event for this subtype. And here, this subtype, the ETV6-RUNX1-like, as you can tell by the name, which means they don't have the ETV6-RUNX1 fusion, but they have very similar gene expression profile as the one with the canonical ETV6-RUNX1 fusion. After our publication here, there are 2 clusters we didn't describe in the paper. One is driven by ZEB2 missense mutation or IGH-CBPE fusion. Sometimes, or in very rare situation, it could be IGH-CEBPB, so other CEBP genes. Here's another subtype, just described in the beginning of this year, which is defined by the high expression of CDX2 and the fusion of UBTF and ATXN7L3. I'll talk about this subtype later. But anyway, using this t-SNP plot, we can visually see all these well-established subtypes and the novel subtypes in a very distinct way. Looking at this figure, you may wonder, the PAX5 alt seems the boundary is not clear. So how can we define if they belong to PAX5 alt or not? To resolve that issue, we used the conventional hierarchical clustering. So as you can see here, the PAX5 alt is clustered in this group. Some of them, they do overlap with the PH-like subtype. But again, using this method, we can at least clearly separate this PAX5 alt group. Within this group, we observed over 70% of them, they carry different types of PAX5 alterations. There are mainly 3 different types of PAX5 alteration. The most common one is PAX5 arrangements with different fusion partners. And the second most common genetic alteration is PAX5 mutations, as shown here. For the PAX5 sequence mutations, some of them, they would show in the biallelic sequence mutations like the PAX R38H or R140L, they're commonly concurrent in the same patient. And for the ones do not have a second sequence mutation, they're commonly concurrent with the deletion of the wild type allele of PAX5, indicating this PAX5 mutations is kind of loss of function. So the need to disrupt the wild type allele before they can develop leukemia. In the PAX5 alt group, we also observed a small number of patients that carry PAX5 intragenic amplification, which is minor in our cohort, but it turned out in the pediatric group, which also takes a decent amount in that. For the PAX5 intragenic amplification, just in case you're not familiar with this genetic event, I'm showing the result here. Here's the whole genome sequencing data. As you can see, within the PAX5 gene region, the exon 2 to 5 have been amplified multiple times. And RNA-seq data is also indicating this result. You can see the exon 2 to 5 is showing with much higher expression level compared to the adjacent exons. Here's another figure showing the RNA-seq level or the gene expression level on each exon, which means using the RNA-seq, we can also faithfully detect this event from the exon 5, exon 2 to exon 5. Actually, if you look closely at the sequencing data, we can identify the event linking the exon 5 back to the exon 2. So I did a quick check of the reads supporting this event from exon 5 to exon 2. You can see there are a large number of reads supporting this abnormal link from exon 5 to exon 2, indicating there's amplification of these 4 exons. but we didn't see that for the other BL cases. All right. For the PAX5 alteration group, we're also interested to see the other concurrent genetic lesions. As I mentioned, we have done whole genome sequencing or exome sequencing to identify the somatic mutations. Among this group, the most common concurrent event is the deletion of CDKN2A, indicating the cell cycle is also disrupted in this subtype. Among this subtype, up to 70% of the cases carry signaling pathway mutation. So that is the signature of this PAX5 outgroup. In addition to the publication, we also try to share this large cohort of data to the research community. That is why we work together with our St. Drew colleagues to develop this web portal. And on this portal, you can explore or you can download the raw read count data or the normalized rate data. You can also explore each sample in this t-SNP plot. You can click each dot. It will show you the detailed information of each sample. Also, you can take a look at the overview of the genetic lesions, like here's the copy number alterations and the sequence mutations, or even the rearrangements. Also, using RNA-seq, we can call the alternative splicing or just the canonical splicing event, which are also visually shown in this on this web portal. Probably most interesting, most informative part is the gene expression across different subtypes, which also you can do on this web portal. You just need to input the gene name. You can check a specific gene expression across different subtype. As shown here, this MAGF10 is significantly highly expressed in the PAX5P8R subtype. Besides the genomic information, we also integrated the survival information here based on the gene expression level. You can tell here, you can check that. Based on the expression level of CRF2, the patient may have different level of a different clinical outcome. So you're welcome to use this website to explore this large data set. Okay, so after the study, we classified this BL cohort into over 20 different subtypes. And this figure shows the distribution of different subtypes across different age group and different risk group, and also different risk groups for pediatric cohort. Again, you can see the high hyperdiploid and the ETV6.1 subtype is highly enriched in the pediatric standard risk group. While the low hyperdiploid, PMT2A, PH-like, and the PH-positive cases, they're highly enriched in the adult group. That kind of explains why the adult cases tend to have worse outcome, because all these subtypes are the high-risk leukemia subtype. So here we're more interested in the 3 major novel subtypes we identified in this study. So as shown here, for the pediatric cohort, the PAX5P8R and the PAX5 out groups are showing similar clinical outcome. But for the adult cohort, you can see the PAX5P8R is showing with much better outcome, compared to the PAX5 out group, while the BCL2 mixed subtype is showing with very poor outcome. All the 6 cases we evaluate here, none of them survived over 2 years. So probably the treatment for this group really needed to be improved. After this study, we also collaborated with another team to evaluate the molecular feature or the clinical feature in the adult cohort, which is relatively pure cohort because all the patients have been treated through this E2-993 clinical trial. Again, here you can see the PAX5 out and the PAX5P8R subtypes are showing with different immunophenotype, and they are classified into different stages of B cell. That probably can explain why we see the difference, the clinical difference of these 2 subtypes. And in this clinical trial, we can see even more drastic difference of the clinical outcome. You can see the PAX5 out is showing with really poor clinical outcome, even worse than the KMT2A rearranged subtype. Another novel subtype I want to briefly talk about here is the CDX2 UBTF. I just mentioned it briefly at the very beginning. We missed this subtype, or we didn't describe this subtype in our previous paper. at the beginning of this year, 4 papers came out to describe this minor subtype, which is showing with high expression of CDX2 and a fusion of UBTF with another gene. This is very interesting subtype because this is showing with very high risk. The patient need longer induction treatment to reach complete remission, or they're also showing with high incidence of relapse, and definitely this is a high risk leukemia subtype. This one is tricky to be detected because it's defined by 2 separate events. So far, we still don't know what is the connection of them. So one event is the CDX2 overexpression. And this is overexpression is induced by the enhancer hijacking to activate the expression of CDX2 because there is a deletion here between the PAN3 and FLT3. If there's no deletion, this enhancer would normally activate the expression of FLT3. But because of that, there's some long distance regular activation of the CDX2 and give rise to the significant high expression of CDX2. And the other event is this short deletion between these 2 adjacent genes and link them together. This fusion is reported as a read-through because they're so close to each other, which is normally missed in our fusion analysis. So in this study, these 2 events, these 2 separate events are considered as the signature of this novel leukemia subtype. Some other interesting information about this subtype is that this subtype is enriched in the AYA group or adolescent young adult group. The median age is around 30 years old, and it's mainly enriched in female patients. And for the reason, we still don't know yet. Okay, next I will take a few minutes to talk about this Paxil P80R subtype because our current lab is still focusing on studying the mechanism of this disease subtype. And also, this is the first leukemia subtype have been described so far driven by one missense mutation. But this mutation is not new. It has been described a long time ago by my previous mentor, Dr. Mulligan, in his Nature paper. This mutation is in the paired box domain, which is a DNA binding domain. There are multiple other mutations in the same DNA binding domain. So at that time, they didn't realize what is the difference of this one compared to the others. So then we look into the copy number variation event of this subtype. And here shows the result. You can see each column represents a sample. Each row is a chromosome. And chromosome 9p region is enriched with a lot of copy number deletions, which is shown in blue color. Dark blue means homozygous deletion, double deletion, and then light blue means 1 copy deletion. You can see these copy number deletions are specifically targeting CDKN2A and a PAX5. Then we combined the copy number variation data and the sequence mutations in this cohort. So you can see here, all these cases, they have PAX5 P80R mutation. 13 of them, they also have a second allele mutation. For the ones who do not have a second sequence mutation, they are observed with a deletion of the wild-type allele, again, indicating this PAX5 P80R mutation is some kind of loss of function of PAX5. We observed over 90% of the cases, they have signaling pathway mutation, especially the RAS signaling pathway. This is the subtype most highly enriched with the RAS signaling pathway. Although it's also involved in other subtypes, but this one is showing this extremely high concurrence with the RAS pathway. To confirm the conclusion that PAX5 P80R is the driver event of this leukemia subtype, we constructed a constitutive knock-in mouse model carrying this PAX5 P80R. As you can see here, the homozygous mice develop leukemia very quickly, normally within 100 days. And the heterozygous one took a little bit longer. And the median latency is around 106 days. And eventually they would develop leukemia carrying the BCL signature, as shown here, the beta-20 positive, CD19 negative. We also carried out some in vivo data to confirm that. So as you can see here, For the Paxil null lineage negative cells, if you do not transfect with Paxil gene, they will not differentiate it to the downstream of the B cell. With the wild type, they can differentiate it to the double positive stage. And same as the other Paxil mutations, but not the Paxil PAR, indicating Paxil PAR is more potent in blocking the B cell differentiation. For the mouse leukemia, we also checked the copy number alterations in the PAX5 region. As you can see here, 2 heterozygous mice, they developed a copy number alteration targeting the PAX5 gene. And even the homozygous mouse, they also have a copy number gain in this region for some reason. This is very similar to what we observed from the patient data. Then we also checked the RNA-seq data of the mouse leukemia. Again, it's very similar. Both the up and down regular genes of the mouse leukemia is very similar to what we observed from the patient data, indicating this is a very feasible mouse model to study this specific leukemia subtype. All right, so with this study, I think we already achieved our initial goal to classify leukemia or the BLL leukemia into different subtypes. And also we developed a very comprehensive BL classification pipeline using RNA-seq data. So using RNA-seq, as we know, we can call the gene arrangements. And with this information, we can identify around half of the BL subtypes. And also using RNA-seq, we can identify the gene expression profile. With the reference data we developed using this large cohort, we can do clustering analysis and distinguish the disease subtype just based on the gene expression profile. Then we can also call the chromosome level copy number alterations, as we have shown before. In addition, we can detect the sequence mutations using RNA-seq data. Combining all this data together, we can reach a relatively convincing conclusion for the BL subtype. So this pipeline is still under active development by our postdoc Zhaohui Gu. With that, I will move on to some ongoing project, which is in our new lab here in City of Hope. After setting up a new lab here, I really want to implement this pipeline in the clinical setting. So we started the collaboration with the researchers and clinicians here in City of Hope. But here, we're mainly treating the adult patients. Here we collected around 100 cases from the tissue bank. And we're gonna do the RNA-seq and then compare the classification results with their clinical diagnosis and to show the accuracy and sensitivity of our method. And meanwhile, we're also collaborating with Dr. Gadana Raka to test our BL classification pipeline in the pediatric cohort. So far, we've already done analysis for almost 100 cases, and now we're preparing the manuscript, and because the result is very promising, I'm very happy about that. On the computational side, we're also developing some bioinformatic tools and pipelines. For example, we're also exploring the multi-omics signatures of each BL subtype. We are doing the analysis on different levels, such as the gene level, exon level, splicing event, or even intergenic level gene expression features. As I mentioned before, we're also using the gene expression data to guide the fusion or structure variation calling. This work is led by the graduate student, Alex. Another part of our research is to connect to the BLL with the normal B cells, because BLL is right from the different stages of B cell, as shown here, from the early pro B to the mature B. And different stages of the B cells, they carry different features, including the VDG recombination and also the cell surface markers. For the VDG recombination, as we know, there could be billions of different combinations of the VDG recombination, which are very good signature or fingerprint to identify different stages of the B cell and BALL. Using RNA-seq data, we can do the digital deconvolution of different cell population, as shown in this figure. You can see here, For BLL, the majority of the deconvoluted result is showing they come from the B lineage, and for TLL, vice versa. The majority of the cell population is from the T cell. Here's a more granular deconvolution with more different B cells. In this study, they showed the KMT2A subtype is more enriched with the ELP cell population, which is a very interesting discovery in this study. Similarly, in our research, we observed that some of the subtype of leukemia belong to a certain or enriched in a certain stage of the B cell. Like the Paxil PDR is probably more enriched in the very early stage of the B cell. And for the BCL2 mixed subtype, it's more enriched in the more mature B cell stages. So definitely different BL subtypes has different signatures. And the other direction is about the VDG recombination events. There was a recent report showing the PAX5 is regulating the VDG recombination by repressing the WAPO gene. Here you can see in the wild-type mice, the V gene used in the VDG recombination is distributed across the V region. But for the PAX5 knockout mice, the V gene used is only the one proximal to the D gene. And the mechanism is PAX5 can repress the expression of WAPO, and the WAPO is the one digesting this cohesin, which facilitates the VDG recombination. Because of that, if there's no PAX5 gene, WAPO gene will be highly expressed, and this will digest the cohesin, and it's hard to form a stable cohesin and to facilitate a diverse V gene connect to the T gene. This figure shows the normal distribution of V gene used in the VDG recombination of the immunoglobulin heavy chain. And in our analysis, we observed that for the PAX5 PDR and the PAX5 out, we did observe a low diversity of the V gene. Mainly the first V gene is enriched in the VDG recombination. So definitely there's some level of defect of the PAX5 gene because of this PAX5 alterations. And this is still an ongoing project in our team. With the mouse data, with the PAX5 PDR mouse lineage in our lab, we're also trying to use single cell sequencing to further delineate the heterogeneity of this disease. So as you can see here, before leukemia, there are different lineage of cells were identified in this mouse. And after leukemia, the B cells are highly enriched, and most of them are pro or pre-B. Here we also did the VDG analysis, as you can see here. All of them originated from the same heavy chain, but they may use different light chain, indicating there are mainly two major cell population within this leukemia. In this mouse, we observed there are two JAK mutations, JAK1, JAK3 hotspot mutations. We hypothesized that these two different mutations are probably the driver of these two different clones of leukemic cells. And this is still an ongoing project. And it's very interesting if we can delineate how the stepwise mutations is acquired in this process. Here's another research initiative we're going to do is to use single cell sequencing to do the disease subtyping. But first, the thing we need to address the cost issue because it's still very expensive. To overcome that, we propose to use the POD strategy, which means multiple samples can be labeled as different hashtag and then put together in one single cell library. Then we can do the gene expression sequencing or the single cell BCR sequencing, and also even the targeting mutation sequencing using the single cell analysis. Then we need to classify the cells into different cell populations. That is why we developed a very clean reference data set from the 1 million immune cells project. With that, we tested one patient. This is already known, belong to the PAX5 out group. As you can see here, all the majority of the cells are annotated as a B lineage, although belong to different B cell stages. Then using the VDG recombination analysis, we identified there are some major clones within this cluster. So we confirm this should be the disease clone. Then with this identified its disease clone, we have a relatively clean gene expression profile inferred from the cells. Then we did the BL subtyping analysis, and all of them are classified as the PAX5 outgroup. It shows the idea, using single cell sequencing, we can reliably and sensitively detect the BL subtype. And again, this is still an ongoing project in our lab. Okay, to summarize, I hope I've already convinced you, BLL is really a highly heterogeneous disease, which consists of over 20 different subtypes. And RNA-seq could be the one-stop platform to classify the different BL subtypes. Actually, over 95% of BLL can be reliably classified using this platform. And because of this granular BL subtyping system, it will give us the chance to develop or discover novel therapeutic strategies for each subtype, such as the TKI for the PH-like cases or the HDAC inhibitor for the MAF2D subtype. And also in our study, we showed that missense mutations can be the subtype defining genetic lesions, such as the PAX5 PADR subtype. And also using the mouse and the in vitro data, we showed that the PAX5 PADR can block the B cell differentiation and lead to the development of leukemia. And last, we just briefly showed that using single cell analysis, we can deliver very clean gene expression profile and the subtyping data of BLL. I think we're run out of time. With that, I would like to thank all my lab members and also like to thank our collaborators here at the City of Hope and also our collaborators from CHLA, especially Godona, which with them, we have very happy and productive collaboration. Also, I would like to thank all the funding support for us to carry out all the proposed research. Okay, I would stop here and take your questions.
Well, thank you so much, Dr. Gu. This was a wonderful talk and so full of exciting information. I'm sure there is a lot for our audience to digest and process, but we did start getting some questions from the audience. So I would invite everybody to put your questions into our Q&A box. And I will start with the questions that we have already received, but just want to say that this was amazing, really fascinating. So many really interesting molecular mechanisms driving some of these new genetic subtypes. And then also great illustration of the power of RNA sequencing and your analytic approaches to characterize these leukemias. So we can start with some technical questions. There are two questions that are not really related, but they're both technical, and they ask details about your analytic approach. So you did touch on advantages of single cell sequencing, but when you're doing your analysis of bulk RNA sequencing, do you have to do cell sorting to be able to cleanly see expression profiles, and especially for your copy number analysis? Or what is the sensitivity, how it depends on the percent of tumor cells? And then I will link that with another technical question. We also had a question from the audience. For your fusion detection, do you only do detection from RNA-seq data, or you do it from DNA sequencing? And if you care to comment about difference between fusion detection from DNA sequencing versus RNA-seq. So those are two technical questions we received.
Thank you, Godona. These are really good questions. And for the first question, the sample preparation, that's actually a very critical step for the RNA-seq analysis. Normally, we would expect highly enriched leukemic cells for the RNA-seq. In our study, the cutoff is 75% for this around 2,000 cases. In that way, we can identify the gene expression more reliable or more representative for the leukemic cell with less contamination. If the blast percentage is lower than 75%, we would do a flow sort to enrich the leukemic cell. But that comes to the next question. If we have some sample with very low blast percentage, like 10%, And even for those samples, even you do the blast or the leukemia cell enrichment, sometimes you cannot reach a decent number of cells, or the cell or the sample quality may not be very good. We encounter some of those samples. That is why we proposed the idea using single cell sequencing with very minimum handling in this process. We can still acquire a very clean gene expression profile of leukemic cell. But again, the cost would have been an issue. That's why we're trying to think if we can get that around using the polar strategy of single cell sequencing. Another question about the DNA, or I can see the Q&A chat box. The long read DNA technology compared to the RNA-seq data. For the fusion detection, I still think it's more effective to use the RNA-seq to call the fusions, because normally the fusions in BLL, for some reason, they're highly expressed because they need to do the work to drive leukemia. So it's like the sequencing rate is highly enriched towards those fusions. It can be sensitively detected. But for DNA, it's equally distributed across the genome. You probably need to sequence more to identify those events. But on the other end, the advantage of DNA is that the material is normally more stable. So there are definitely some advantage of that. Considering the long read DNA, it could be beneficial to answer some questions like IGH-DAX4. I'm not sure if the audience is familiar with that. Another paper, they use the PacBio long read sequencing to identify the IGH-DAX4 rearrangement to see which IGH region is rearranged with DAX4. Because normally we assume the activated the IGH region, but actually the result is the opposite. The non-rearranged IGH region is actually is connected with DAX4. Because in their study, they showed that extremely high DAX4 is detrimental to the cell growth. So it's just a decent level of activation of the DAX4 from the non-rearranged IGH region is actually the one leading the rearrangements. So definitely the long-read DNA sequencing can bring more insights to understand the mechanism of the genetic events.
Great, thank you so much. Excellent explanation. You give us a lot of really interesting new information about PAX5 driven subtypes, including P80R and PAX5 alt. But then we also see PAX5 mutations and often same types of alterations as secondary abnormalities in a variety of different subtypes. So what do you think is the difference between PAX5 sometimes driving and being the subtype defining change, and sometimes just being cooperating change? Is it in the mutation type? And are you seeing differences in mutation types, or timing, or the fact that you have to have if you get biallelic 2 mutations that pushes in leukemia into that PAX5 driven subtype? Have you been able to decipher how can PAX5 mutations be both primary and secondary changes?
Yeah, this is a very intriguing question. Thank you, Kadona. We've been focusing on the PAX5 alt or PAX5 P80R subtype, but still it's really hard to claim, okay, this PAX5 mutation is the driver. Sometimes we do see PAX5 mutation could be the secondary. Like for the PH-like subtype, we observed the PAX5 V26G missense mutation is as a secondary event, because that can be, that mutation can have lower MAF, indicating that just exists in a small number of the cells. But some of the mutations were pretty confident they should be the driver, like the P80R or like the R38H or R40L. Some extra evidence can corroborate this conclusion is the germline mutations. Like for R38H, there are some reports showing some familial cases. In their family, they carry the R38H, and their family tend to develop BLL. And we check their data. They do confirm the signature of the PAX5 out group, indicating this could be a driver event or even can exist as a germline susceptibility to develop this subtype leukemia. Another observation we made in the study is that PAX5 deletion tend to be the secondary event. It can coexist with, for example, ETV611, which commonly see with PAX5 deletion. And definitely this is a secondary event. So we cannot claim that is also a driver for PAX5 out. But PAX5 out subtype is not that well defined. I do admit that. That is why we still need a lot of effort to study this subtype. And hopefully in the future, we can provide a better or a full picture of the PAX5 out group.
Oh, great. We might easily have some people in the audience who might be interested in collaborating with you in future studies related to PAX5 out and other BLL subtypes. Unfortunately, we ran out of time, but this was a really wonderful and fascinating talk. So thank you again for joining us. Thanks to all the audience for joining.
Thank you. It's really my honor.
Yes, and there were some questions that did not get answered. So I did a copy of those and I will send them to Dr. Gu and I don't know how we can allow those questions to be answered, but we'll try and find some mechanism. I also wanted to remind those that we do have our annual meeting in St. Louis from July 31st to August 3rd. And please go to the registration site at consortgenomics.org. So thank you again for everybody for joining us today, and I look forward to seeing you for the next one.
Thank
Bye,
you,
everybody.
Everyone.
Bye.